So I'm going to discuss how to do genetic engineering. Let's say we found a gene that we really like. Like, for example, we want to produce insulin on a mass scale. So we'll get a human cell, we'll get the nucleus, we'll look at the DNA sequence. We'll identify which part of the DNA sequence, the specific gene that produces insulin. So if this is the DNA sequence, we will cut this section that will produce insulin. And to cut the ends, we use something known as a restriction enzyme. So voila, in the green, we have a specific gene that produces insulin. We will then go on to a bacteria cell, and the bacteria cell have something known as plasmids. So the plasmid is seen as this black circle over there. And what we have to do, we have to open up this plasmid. And using the same restriction enzyme we use to cut this gene of insulin, we will open up the plasmid with that restriction enzyme. This just makes it easier just to glue back on. We then get the gene for insulin and we put it inside the plasmid. Now to glue it on the ends, we use something known as a ligase enzyme. Now this is now known as a recombinant plasmid because it contains a specific gene, and we put this back into the bacteria cell. Now we have a bacteria cell that has the recombinant plasmid, and we can mass produce this bacteria, which also makes copies of this insulin. Later, the insulin needs to be extracted and purified before it's used in human beings.